Lou is a young scientist who has been very successful in publishing. If you touch his picture at the website, we will see that 2012 it was a perfect year for him. He published in New England Journal, Lancet, Circulation, and I believe is in genetics, Nature Genetics, I believe. It was selected papers that were posted in the site just to give you an idea about him. He was talking a few minutes ago with me about the interactions in the nutrition department. Although he has been working with Frank Sachs, Walter Willett, Eric Green, he is an independent scientist with focused on genetics, obesity, and cardiovascular disease in general. He is very welcome to start his presentation, and I hope you enjoyed, as I did a few weeks ago.
Thank you for the nice introduction. Good morning. So I also like to take this opportunity to thank the organizer, especially Dr. Sandra Fugers, for the invitation. My great pleasure being here. So as Sandra just introduced, my research field is kind of combination of nutrition epidemiology, genetic epidemiology, and interactions between the environment and genetics. So today I'm going to show you some relatively fresh data we got in the past around 2 years about the gene-environmental interaction. For the environmental factors, mainly lifestyle factors and dietary factors in relation to obesity. So this is an overview of my talk. First, I'm going to talk a little bit about background, epidemiology, risk factors, and genetic findings. In the second part, I will focus on the gene lifestyle interactions, with focus on physical activity, TV watching, sedentary lifestyle. In the third part, I will talk about gene data interactions in cohort studies. And in the last section, I will address the gene data interactions in randomized clinical trial. Background. So obesity, simply speaking, is a condition of excess accumulation of body fat. So obesity is defined on the basis of body mass index, BMI. So BMI greater than 30 is termed as obesity, greater than 25 is termed as overweight. So this figure shows the prevalence of overweight, the upper figure, and obesity, the lower figure, over the world. According to the data published in Lancet 2006, there was 1.1 billion adults, 10% of children are overweight or obese over the world. And it was estimated that by year 2030, there will be 2.1 billion overweight and 1.1 billion obesity. So obesity is a risk factor for a variety of metabolic diseases, such as diabetes, cardiovascular disease. So the epidemic of obesity has become a major public health and also economic burden. So this slide shows the causes or risk factors for obesity. Some risk factors are very well known. For example, just before the coffee break, it was mentioned that low physical activity, sedentary lifestyle was related to not only cardiovascular disease, but also obesity. And the dietary factors, the high intake of calorie, high consumption of sugar-sweetened beverage have also been related to increased risk of obesity. So in addition to these established risk factors, some other so-called non-traditional risk factors include lower social status, air pollution, psychological factors, depression, peer pressure, and very recently, gut microbiota are also. Okay. Thank you. Very recent gut microbiota also related to a risk of obesity. So in addition to these environmental factors, the genetic research in the past decades has clearly indicated that genetic factors also contribute to the development of obesity. So according to the classic genetic research, the twin studies, family studies, as high as 50 to 60% of the variance of obesity could be explained by genetic factors. And in the past 5 or 6 years, there are about more than 10 studies, so-called genome-wide association study, GWAS study, have been published. This study together identified more than 30, actually 32, genomic loci associated with BMI, associated with obesity risk. So this figure is so-called Manhattan plot, which lists all the identified loci associated with BMI and obesity risk. Most of the genetic effects are very weak, very moderate. So the genetic factors, environmental factors can affect obesity directly. They can also interact with each other in determining the risk of obesity. So as for the relation between genetic factors and environmental factors, Dr. Francis Collins, the director of NIH, has a very famous comment. He says, genetics loads the gun and environment pulls the trigger. So I think this is a very beautiful translation of the scientific term, gene-environmental interaction. Oh, sorry. So one line of evidence supporting the potential interaction between genetic factors and environmental factors is from the observation of the temporal change of obesity in the US population. As you can see that the prevalence of obesity is very low in 1970s and start to rise since middle 1970s. And the increasing trend slowed down and stopped around 2000. So afterwards, the prevalence of obesity kept at a relative stable level, about 30-35% in the US men and women. So what that means? That means in this population, 60% remains not obese, and 30% remains lean with BMI less than 25. Even though the whole population continues to be exposed to the so-called obesogenic environment. That means there is considerable diversity in the population in response to the obesogenic environment. This is actually the gene-environmental interaction. Another line supporting the potential link between the environmental factors and genetic factors is the observation that most of the obesity genes was found to be highly expressed in brain, especially hypothalamus. Hypothalamus is the regulatory center for the balance of energy intakes and energy expenditure. So it's a key regulator for the dietary intakes and the energy expenditure, physical activity. So this observation provides strong biological basis for the potential interactions between genetic factors and environmental factors, especially dietary intakes, physical activity, in determining obesity risk. So in the past few years, our group has performed several studies to address the gene-environmental interactions. For the environmental factors, we focused on the factors heavily affect the balance between energy intake and energy expenditure. For example, physical activity, sedentary lifestyle, and the dietary intakes, for example, sugars and beverage consumption. So in the next part, I will address the gene-lifestyle interactions. So to detect the interactions between genetic factors and environmental factors, about 3-4 years ago, we formed a consortium called Diet Gene Consortium. This consortium consists of 3 large prospective cohorts: the nurses' hair study, the health professional follow-up study, and the women genome hair study. The nurses' hair study, all the participants are women. They were followed since 1976. And health professional follow-up study, all the participants are men, physicians. They were followed since 1986. And the women genome hair study, again, all the participants are women. They were followed since 1992 to 1995. So among these cohorts, we have nearly 7,000 women from nurses' hair study, 4,400 men from health professional follow-up study, and nearly 22,000 women from women genome hair study have a genome-wide scan available. So we collected, in all these 3 cohorts, we collected the information of lifestyle and dietary intakes by using follow-up questionnaires and food frequency questionnaires. All these questionnaires have been very well validated in this cohort. For the genetic factors, we evaluate the overall genetic susceptibility by considering all the genetic loci identified from the GBAT study, as I just mentioned, in total 32 obesity genes. So we calculate a score called genetic risk score, which is calculated by summing up the number of risk allele of all the 32 obesity genes. So 1 point of the genetic score is roughly equal to 1 risk allele of obesity gene. And the higher the genetic score, the higher BMI, the higher risk of obesity. So this is the paper we published last year in Circulation. In this study, we test the interactions between the genetic score, the marker for the overall genetic susceptibility, for its interactions with physical activity, with TV watching in relation to BMI and obesity risk. So TV watching is the most common sedentary lifestyle in the United States. So in previous epidemiology studies, TV watching and the lower physical activity have been independently related to obesity risk. So in this study, we found significant interactions between the genetic score and the physical activity, TV watching in relation to BMI. So this figure shows the results, the genetic effect according to the quintiles of physical activity. The genetic effect is presented as the difference in BMI associated with 10 points of genetic risk score, which corresponds to 10 risk allele of obesity gene. Hope it's clear to you. So as you can see that the genetic effect was stronger in those with low physical activity as compared with those with higher physical activity. In other words, the higher physical activity attenuated the genetic effect on BMI. The test for interaction between the genetic score and physical activity was statistically significant. The p-value is less than 0.001. Also, we found significant interactions between the genetic score and the TV watching, which is a marker of sedentary lifestyle. We found that the genetic effect was stronger in those with spent more times on TV watching, greater than 41 hours per week, than those spent less hours, less times on TV watching. In other words, the sedentary lifestyle, TV watching, may exaggerate the genetic effect. So as I just mentioned, the TV watching and the physical activity are independent risk factors for obesity. In our cohort, the correlation between these 2 variables, the correlation coefficient is 0.03. That means very weak correlation. So we also test the joint interactions between of physical activity and TV watching with genetic risk score in relation to BMI. To this end, we divide the participants into 9 groups according to the 3 categories of TV watching and 3 categories of physical activity. For this group, who had the lowest physical activity and the highest levels of TV watching, this is actually the most sedentary group. So the genetic fact, again, the genetic fact is presented as difference in BMI associated with the 10 points of genetic score. A genetic fact is 2.5 units. In this group, which has the highest physical activity and the lowest levels of TV watching, this is actually the most active group. The genetic fact is 0.2. So in other words, the genetic effect in this most active group was almost completely eliminated. Actually, as compared with the most sedentary group, in all other groups, the genetic effects are attenuated. For example, in this group, who had the highest physical activity, but also highest levels of TV watching, this group are very active, but also like TV watching. So in this group, the genetic effect is 1.0 units difference in BMI. So as compared with the most secondary groups, the genetic effect in this group was attenuated by 60%. So if you really like TV watching, so the best recommendation is to watch TV on treadmill. So next, I'm going to talk about the gene-data interaction in the cohort study. So this is the paper we published last year, September 21st, in New England Journal of Medicine. In this study, we tested the interactions between the genetic score or genetic susceptibility with sugar-sweetened beverage consumption in relation to BMI and obesity risk. Sugar-sweetened beverage are beverage with added sugar, which include soda, other carbonated soft drinks, juice drinks, sports drinks, energy drinks, and sweetened milks, sweetened tea or coffee. So this figure is from the paper published in Nature 2012, which shows the level of sugar-sweetened beverage consumption over the world. The dark color represents the high consumption. As you can see that the United States has the highest consumption of sugar. Actually, sugar-sweetened beverage is the #1 source of diet-added sugar, which contributing about 36% of the added sugar in from diets, and also one of the major source of calorie intake. So this figure shows the temperature change of sugars and beverage consumption in the United States in children and young adults. The red line represents the sugars and beverage intake. As you can see that the consumption of sugars and beverage started to increase from late 1960s and stopped around 2000s in both children and adults. Remember the last figure just showed you the temperature change for the obesity. So this temperature change pattern is very similar to the temperature change pattern for obesity. And so this data suggests that the increase of sugars and beverage intake may contribute to the development of obesity in the same period of time. Actually, in the epidemiology studies, especially the large-scale prospective studies, there's a huge body of evidence has clearly shown that the high consumption of sugars and beverage is related to weight gain and obesity risk. So there are many mechanisms has been proposed to explain the link between SSB intake and obesity. So one prevailing explanation is that as compared with solid food, the calorie intake from sugars and beverage may lead to incomplete compensatory reduction in the subsequent calorie intake. So the long terms of a high consumption of sugars and beverage may lead to the broken of the balance between energy intake and energy expenditure, and finally lead to the development of obesity. So in this study, we tested the interaction between genetic susceptibility and the sugars and beverage intake in relation to BMI. So to this end, we divided our participants into 4 groups according to the consumption of sugars and beverage from low to high. The lowest group consumed less 1 serving per month sugars and beverage, followed by 1 to 4 serving per month and 2 to 6 serving per week. The highest group consumed greater than 1 serving per day sugars and beverage. So we showed, here I show you the genetic fact in the groups, in the 4 groups with different intakes of sugar and beverage. Again, the genetic fact is presented as the difference in BMI associated with 10 points of the genetic score. We, in the first cohort, the nurses here study, we found significant interactions between SSB intake and the genetic score in relation to BMI. As you can see that the genetic effect was stronger in those with higher SSB intake than those with lower SSB intake. A test for gene-data interactions points that there are 8. In the second cohort, the Harris professional follow-up study, we found very similar interaction pattern. The genetics effect was stronger in those with higher SSB intake than those with lower SSB intake. Again, the test for gene-data interaction is significant. If we combined the 2 cohorts together, the gene-data interaction becomes more significant. And next, we try to replicate our findings in the 3rd cohort of our consortium, the Women Genome Hair Study. Here is the result. So we found exactly the same gene-data interaction pattern in the 3rd cohort. So again, the genetic fact is stronger in those with higher SSB intake. So this data taken together provides strong evidence for the potential interactions between the genetic score or genetic susceptibility with sugars and beverage consumption in relation to BMI or obesity risk. And the high consumption of sugars and beverage may exaggerate the genetic effect on obesity. So these findings are very interesting. However, they were all performed in the observational settings. So we know that observational studies are subject to the various bias, various limitations. For example, confoundings, reverse causation, this is the actual particular issue in the cross-sectional analysis. And the measurement errors, especially lifestyle factors, data factors, the measurement error is huge in observational study. And more important, in observational study, we cannot directly evaluate the response to the environment factors. And such information is very important regarding the future development of interventions to reduce obesity. So an alternative, actually better study design is a randomized clinical trial. As compared with observational study, randomized clinical trial has several major strengths. For example, in RCTs, randomization minimizes confounding. And the prospective design reduces the risk of reverse causation. Because the intervention are prescribed, the measurement error for the exposure is very, very low. And more important, in the randomized clinical trial, we can directly evaluate the response to the environment factors. So we recently also tested the gene-data interactions in randomized clinical trials. So this is the randomized clinical trial. We tested the gene-data interaction, the pound loss trial. The preventing of the overweight using novel dietary strategy trial. This is the NIH-funded study. The PI of this trial is Dr. Frank Sachs. He's professor at Harvard Medical School and Harvard School of Public Health. The original paper reporting this trial was published in New England Journal of Medicine 2009. This is a 2-year diet intervention trial consisting of 811 overweight or obese participants. So this study tested 4 diets varying in macronutrient intakes. Also, all these diet groups has a lower calorie intake. So these are the 4 diets tested in this trial. Diet group 1, 2, 3, 4. They are different in the macronutrient intake, dietary fat, protein, and carbohydrate intake. So this study design is very smart, so we can compare test the dietary effect in different ways. For example, if we are interested in dietary fat intake, we can combine the first two diet groups together with 20% of calorie from dietary fat as a low-fat group. And we can combine diet groups 3 and 4 together as a high-fat group with 40% calorie from diet fat intake. So if we are interested in protein intake, we can combine the diet group 1 and 3 together as a low protein groups with 15% of calorie from protein, and combine the diet group 2 and 4 together as high protein groups. We can also compare the different dosage of carbohydrate intake. So this is a paper we just published in 2011. In this study, we tested the gene-data interactions with addressing a gene, RS1 gene. This gene encodes insulin receptor substrate 1 protein. This is the key player, key factors in the insulin signaling pathway. This gene is very special because in several genome-wide association studies, it was found that the genetic effect, genetic variant in RS1 gene was related to various metabolic traits. For example, type 2 diabetes, insulin resistance, hyperinsulinemia, body weight, and lipid. So we test, also interesting, in a small randomized clinical trial, it was found that the RS1 genotype interact with dietary intake, especially carbohydrate intake, in relation to insulin sensitivity response. But this is a very small and short-term intervention study. So we genotyped the RS1 genotype in the PANLOS trial. This figure shows the change or the reduction of the markers of insulin sensitivity, the fasting insulin level, the HOMA-IR levels, according to the genotypes of RS1 gene in the 4 data groups. Data group 1, 2, 3, 4. The blue bar represents the individuals with the risk genotype, the CC genotype. The red bar represents the individuals carrying the other genotype. So we found significant interactions between the RS1 genotype and the carbohydrate intake in relation to the 2 markers of insulin sensitivity. In the data group 1, which has the highest carbohydrate intake, you can see that the individuals carrying the CC genotype, the risk genotype, has a greater reduction in insulin level, in HOMA-IR level, as compared with individuals carrying other genotypes. In the data group 4, which has the lowest carbohydrate intakes, we observed opposite direction genetic fact. Individuals carrying the other genotype has greater reduction in these 2 markers of insulin sensitivity. So this data support the potential interaction between carbohydrate intake and the RS1 genotypes in relation to the insulin sensitivity response. So individuals carrying the CC genotype may benefit more in reduction of insulin levels, in resistance levels, by taking low-calorie, high-carbohydrate weight loss diet. So after publication of this paper, we tried to find the additional randomized control trial to further validate our findings. So different from the observational study, it's extremely difficult to identify, to find a study with identical designs for randomized trials. But fortunately, we found another study with a very similar design, the direct trial. The dietary intervention randomized control trial. This is a real-based study. The PI of this study is Dr. Iris Shea. The recent paper reported this trial, again, was published in New England Journal of Medicine, 2008. Similar to the pound loss trial, this is a 2-year intervention trial consisting of 322 overweight obesity subjects. And slightly different from the pound loss trial, this study tested 3 diets, the low-fat or high-carbohydrate diet, low-carbohydrate or high-fat diet, and the Mediterranean diet. So this is the university, the study was carried. The Ben Gurion University, this is located in a small, beautiful city in Israel. Oops. And this is the very popular animal in this area. So this table compares the 2 trials. The direct trial, the pound loss trial, you can see how similar they are.
Pessoal, bom dia. Para a gente não demorar muito, vamos tentar fazer uma reunião um pouquinho mais enxuta hoje e que a gente possa ter espaço para trocar ideias.
Okay. Okay, let's continue. So you can see the 2 trials are very similar in study design. In both trials, the participants are overweight or obese adults. In both trials, this 2 years intervention, and even the diets tested in the 2 trials are slightly different. Both trials allow to compare low versus high fat or high versus low carbohydrate. So recently we genotyped the same RRS1 gene SNPs in the direct trials. And this figure shows the result. Again, the figure shows the reduction of the fasting insulin level and HOMA levels in response to the diet intervention. And the blue bar represents the individuals carrying the CC genotype, and the red bar represents individuals carrying other genotype. As you can see that in the diet group with a high carbohydrate intake or low fat intake, individuals carrying the CC genotype had great reduction in the both markers as compared with those carrying other genotype. In other diet group, the low carbohydrate diet groups, individuals carrying other genotype had a great reduction in fasting insulin level, in insulin resistant levels. So the test for interaction between the diet intervention and the SNP, genetic variants, again, are statistically significant. The p-value is 0.003 and 0.008 for the 2 markers. So if we compare this result with what we observed in the pound loss, you can see that the gene diet interaction pattern are very similar in the 2 trials. So the replication data really convince us that the potential interactions between carbohydrate intake and RS1 genotypes are likely to be real. So as a summary of my talk, the analysis in both observational study and randomized trials have shown that the genetic factors may interact with environmental factors in affecting obesity risk and related metabolic traits. And the gene-environmental interaction study in randomized controlled trials may provide complementary and novel information that couldn't be obtained from observation settings. And the research of gene-environmental interactions is still facing many challenges, but holds great promise to improve our understanding of obesity. So I would like acknowledge my colleague at Harvard School of Public Health, Harvard Medical School, the Pennington Biomedical Research Center, and the investigators from the direct trials. So thank you for your attention.